Hi, so today our topic is going to be vaccines. So, first off, you have to know that vaccines are basically there to stimulate your immune system and fight against pathogens, right? And they are mostly involved in your active immunity. And the main goal of vaccines is to generate memory B and T cells. Now, the reason why this is very important is because when someone is exposed to an infection the first time, they don't mount a very strong immune response. But what happens is that upon re-exposure to that same infection, because they have made memory B cells and T cells from the previous infection, they have a much faster and stronger immune response. So this is our goal with vaccines, right? So we want to give them antigens that represent the infection itself, and that can lead to the generation of memory B and T cells. And the next time when they are exposed to that infection, they have a much stronger and a longer lasting immune response, right? So the main goal is to mimic the infection without causing the disease, right? Because most of our vaccines are basically killed off, or they are just pieces of antigens of that infection, right? So basically they can't cause the disease, but we want them to mimic the infection. So what does that lead to? That leads to a faster, stronger, and a long-term immune response upon re-exposure. Why? Because now you have memory B and T cells. And just a general high yield rule that you have to know regarding vaccines is that the closer the vaccine resembles to the pathogen, the stronger the immune response is going to be. And this is going to become evident when we are going to be learning about the different types of vaccines. So first, we have live attenuated vaccines. So these are weakened but living organisms, right? So they are weak, they cannot cause the infection, but they still are living, right? And that is because they have the ability to replicate. Now, they strongly mimic the natural infection, right? The only thing that they can do is that they cannot cause an infection, right? But other than that, they have the ability to replicate, and they're just weakened to the point where they cannot cause any harm to the body. Now, this induces humoral plus cellular immunity. So you produce antibodies, plus you also cause T cell proliferation, and that leads to the production of your memory B and T cells. And that is why you have a strong and long lasting protection. And this is one of the advantages of it, that it usually requires fewer boosters. So just in general term, boosters are vaccines that you give again and again, time after time. Basically what happens is that, let's say that this is a graph, right? And this is when you injected a vaccine. So your immune response is high, and let's say it keeps going on through your life. But as you go on, the immune response, it starts fading away. And that is one of the reasons why after some time you again give them a booster where it will again rise up. And this will just keep on continuing. And the main reason behind this is that the immunity does not wane off, right? But with live attenuated vaccines, since the immune response is so strong, that your graph will look something like this. So you see, your immune response does not wane off. So that's why they require very few boosters. But there is a disadvantage of it. It is mostly avoided in immunocompromised and pregnant patients because of a single reason. Why? Because their immune system is weakened. Yes, pregnant patients have a weak immune system. Now, why is this important? The reason being because, like I said before, they still have the ability to replicate. So if your immune system is weak, the virus still has the ability to replicate. So it will replicate, and that can cause reinfection, right? So that is why we avoid it in immunocompromised and pregnant patients. Now, some of the examples of live attenuated vaccines are going to be MMR, so measles, mumps, rubella, varicella, which is basically your chicken pox, rotavirus, which can cause acute viral gastroenteritis in children, especially polio, and BCG. All right, next we have inactivated vaccines. So in this, the organisms are killed off, right? So they do not have an ability to replicate. But the structures, or the antigenic structures, they are preserved, right? So mainly the humoral immunity is induced in this vaccine, in inactivated vaccines. And because they do not have the ability to replicate, they have no reversion to virulence, right? So they cannot reinfect you. And it is safe in immunocompromised and pregnant patients. But there's a caveat to this, is that you require multiple boosters and doses. Why? Because again, the same example, because it does not mimic the natural infection as close to live attenuated vaccines. That is why your immune system will wane off, and that's why you have to keep giving them multiple boosters throughout life, right? So weaker and less durable response than live vaccines. And some of the examples are going to be polio, hepatitis A, rabies, influenza. And by the way, polio is intramuscular polio, not the oral polio. Next, we have subunit and recombinant. So subunit is going to be only specific pathogen antigens are going to be used in this. So basically, let's say that this is your virus, right? So this is your virus. And instead of using the whole virus, right, you only use the receptor proteins, right? Or you use the antigenic proteins that are present on it, right? You don't use the whole thing. On the other hand, recombinant is when the antigen is produced using genetic engineering. So it's mostly seen in virus, right? So you take a virus antigen and you put it inside a yeast cell, and the yeast cell will start producing that antigen, and you can use that as a vaccine. But here's the caveat. So no live organism is used, right? You have a generally targeted immune response, and it is safer. Why? Because again, they don't have the ability to replicate. And some examples are going to be hepatitis B vaccine, HPV, human papillomavirus, and recombinant zoster vaccine. Next, you have polysaccharide and conjugate vaccines. So polysaccharide are going to be vaccines in which you use the capsular polysaccharides that are present mostly on bacterial surfaces. You use them, and that leads to a T cell independent response. Now, can you tell me why it's a T cell independent response? Exactly. The reason being because T cells require a protein structure. If there is no protein structure, the T cell cannot recognize it. And this is the reason why they have a weaker response. And you don't have formation of memory B or T cells, and it is ineffective in children. So one of the example is going to be PPSV23, which is basically for strep pneumo, right? So streptococcus pneumonia. Conjugate, on the other hand, is going to be using the polysaccharide portion plus adding a protein part. Why? Because the protein is going to be responsible for the T cell dependent response. So again, if you have a protein structure, the T cell is going to recognize it and it will mount a stronger immune response. But if you don't have a protein structure, that leads to a weak response, right? So in conjugate vaccines, you have a stronger response and formation of memory B or T cells. And some of the examples are going to be Haemophilus influenza, right, meningococcal conjugate vaccine, and DTaP, which is basically a vaccine used for pertussis, right? So Bordetella pertussis. Okay, next we have what is known as toxoid vaccine. So toxoids are basically inactivated bacterial toxins. So if you have studied micro, you do know regarding your exotoxins, right? So exotoxins are basically very dangerous molecules that are produced by the bacteria that can cause very severe damage to your body, right? So what happens is that toxoid vaccines are going to be inactivated bacterial toxins. So the toxicity part is removed, but the antigenicity is preserved. So the antigen function is preserved, but the toxicity, the harmful part, is removed. Now what does that do is that it produces antibody against the toxin, not the bacteria. Why? Because let's say this is a bacteria. So the bacteria has a different antigenic structure, right? So it has a different receptors or different antigens on it. So the bacteria produces an exotoxin, and that exotoxin goes out and causes harm to your body. So if you're producing an immune response against that toxin, that will not be the same as producing an immune response against the whole bacteria, right? So that is one of the reasons why this is not effective in eradicating the bacterial infection, but rather the harmful effects of the toxin. So one of the most high yield examples is going to be tetanus, right? So tetanus is obviously Clostridium tetani, and you will mostly see this in patients who have had road accidents or any kind of motor vehicle accident in which they have been on contact with the surface, right? And whenever we have these patients in the hospital, we immediately inject them with tetanus vaccine, right? The reason being because if the treatment is not given in time, that can lead to a whole body-wide spasmic reaction, right? Where all of their muscles will spasm up, and it's a very painful condition, right? Nobody wants to be in there. All right, so next is going to be diphtheria. So diphtheria is basically Corynebacterium diphtheria, right? One of the most highest yield diseases that it causes is known as pseudomembranous pharyngitis, right? So when you look at their pharynx, right, it will look very whitish color, right? It will be basically pseudomembranes that have formed on top of the pharynx, right? And it's a very painful condition in which their breathing is compromised, right? And along with that, Corynebacterium can also cause myocarditis. So you can imagine that we need a vaccine for this, right? And especially a toxoid vaccine. The reason being because diphtheria produces an exotoxin that causes what is known as ADP ribosylation, right? And that can lead to cell death and necrosis, right? And these toxoid vaccines, they require boosters, right? With the same concept, because the immunity wanes over time because you are producing an immune response against the toxin, not the bacteria itself. Okay, last we have mRNA vaccines, and this is one of the more recent ones, right? So what happens is that there is a synthetic mRNA that we make in the lab, right? And then we inject it into the host cells, and what the host cells do is that they produce the pathogen antigen. Now, again, they are not producing the pathogen itself, they are only producing a part of its antigen, and then that leads to an immune response. And this leads to both humoral and cellular immunity. And again, no live pathogen is used, so obviously no risk in immunocompromised or pregnant patients, and it is highly effective. It can cause some local or systemic reactions like myalgia, fever, pain in the body, pain at the injection site. But other than that, one of the more rare serious complications is myocarditis. But again, that is very rare. And the most important vaccine, and I believe you're all familiar with this, is the SARS-CoV-2 mRNA vaccine. So this is the COVID-19 vaccine that recently we have all been injected with, right? Maybe. So it was named as the Pfizer, Biotech, or Moderna vaccine,